All right, hey everybody. So we're going to continue going over the notes today. Before I left, before the weekend, we covered the first part of chapter 10. So remember, we said that as a cell continues to grow, it has two options. It can either choose to divide or it can choose to die. And so if you'll follow along with my mouse there on the screen, we're going to walk through the notes, make sure that you're filling in the blanks as we go. You may actually need to pause this. That way you can actually fill in the blanks, write in stuff, make sure things are spelled correctly. That it would just prevent, you could pause it, do whatever you want. I don't know. You can go backwards and rewrite stuff, rewatch this. If you go home, it's on YouTube, so you can watch it there too if you want to, if you're just extremely bored. But we talked about cell division. We said that a cell has limits to its size that it can grow, and those deal with information overload, meaning that the nucleus gets run down with information. There's too much data for it to process, and then material exchange in the cell is going to slow down as well. We compared that to traffic in large cities versus traffic in French camp. Obviously, the larger the surface area, the more traffic and volume you have in there, the slower you're going to move from place to place. Whereas if it's smaller, you have less traffic, then you're going to move much quicker. So our solution to this problem with the information overload and material exchange was cell division. So there's two different kinds of division here. You've got asexual and sexual reproduction. So I think some of you got to write this down on Friday, but we didn't get to talk about it a little bit. Asexual reproduction, this is going to be happening at the cellular level. It's production of genetically identical offspring from a single parent. So you have one parent cell, and it's going to produce two new identical daughter cells. This version of reproduction is faster, but it provides less genetic variation, meaning that you're going to make a numerous amount quickly, but in the end, it's not as resilient. It's easier to wipe out that population versus one that's not done asexual reproduction. In sexual reproduction, you have the fusion of two separate parent cells. So you have two cells coming together to make one, versus asexual, you have one that splits to make two. And so you have two cells here, they come together, they fuse one new cell. And the offspring will inherit genetic information from each parent. So in humans, for example, you give half of your DNA through your sex cells, they combine with another, and then the offspring ends up with the correct number of chromosomes. This is a slower process, but it provides more genetic variation and resilient populations. And so it takes more time. The number or volume of organisms produced is slower than, let's say, bacteria growing on something by the hundreds over the course of a couple of hours. And this is going to take more time, depending on the organism, months, or however long it is. So it depends on the organism. Every organism has a different gestational period. Let's come back up here and look at chromosomes. I hope you got that. Remember, if you miss something, pause it or go back. I'm just going to keep rolling through this. So chromosomes, these are the things we're talking about when we talk about division and what's passed on. This is what's in the nucleus, and part of what the nucleus has to go through in order to make the proteins, or give the instructions for making the proteins needed for the cell. And so the structure chromosomes here, you've got DNA, which you're familiar with. It's in the spiral helix that you're all familiar with. And it's going to be wrapped pretty tightly around these histone proteins. And so when it does that, it makes a nucleosome. And then when you combine and put these nucleosomes on top of each other in coils here, that is going to be what makes our chromosomes. Most prokaryotic cells, these are small single-cell organisms, contain a single circular strand of DNA. or DNA chromosome that contains the cell's genetic info. So they don't have a nucleus that holds the information. They're going to have a single strand, usually in a circle, located in the cytoplasm, and that will be where they get their information from. Eukaryotic chromosomes have more DNA than prokaryotes, and it will exist in multiple chromosomes, not a single circular strand there. In its simple state, it is chromatin. So for the majority, or at least the early part, until the cell starts to get ready to copy and divide, actually all of interphase, it's chromatin. And then when we move into mitosis is when it condenses to form chromosomes. But it exists as chromatin, which is an uncoiled version. It's easier to go through and read and make things from. But during division, it's going to condense to form chromosomes. Chromatin is not visible to the naked eye under the microscope, at least a light microscope, but chromosomes are. And so whether you can use these two interchangeably, whether it's chromatin or chromosomes, these are made up of DNA and histone proteins. And if you combine DNA and histone proteins, you get nucleosomes, and that's what makes up our chromatin or chromosome units. For the, let's look at prokaryotic. For the prokaryotic cell cycle, specifics not entirely known. That's not entirely true. We will do a different unit on this if we have time before I leave, where we'll get into more specifics about how the strand here will attach to certain places, how it'll swap DNA with other single cell organisms. That's how it'll help it to evolve. That's how viruses mutate or bacteria mutate and things. And so we'll look more at that later. But what we're going to look at is this process here. What this is called, and it's not on here, so you'll need to write that somewhere over here. It's called binary fission. If you can't spell that, that's binary, binary, and then fission, fission. This is similar to the eukaryotic cycle, the fact that the cell is going to grow, replicate DNA, and the DNA is going to move to opposite sides of the cell, and then the membrane will pinch in and form two new daughter cells. And so you get the cell membrane and your circular strand of DNA. As the DNA, after it duplicates, it's overlapping, and then it's going to start to pull apart. As it pulls apart, the membrane begins to pinch in, and then you end up with two new cells. So in prokaryotic cells, this is going to be binary fission, not mitosis, binary fission. Make sure you get that down so you don't mess that up on our test. For the eukaryotic cell cycle, we're going through interphase. And so this is where the cell spends the majority of its life. So if we're going to scroll down and look real quick, if this is the life of the cell, this small portion in green is mitosis. The rest of the cell's life, it spends in interphase. So this is a pretty lengthy, think of everything we've studied so far with the cell and how things move, photosynthesis, respiration. All of that happens during interphase. Interphase is represented by three different phases, G1, S, and G2. G1 phase is cell growth. The cell does most of their growing during this phase. They increase in size and they synthesize new proteins and organelles. So at this point, we know they've just come out of a previous division, so they're still small relatively. So they're growing both in physical size and the amount of things that they have in them. They're producing new proteins, organelles. Those organelles have functions that they're working on. And so this is where most of your normal process in a cell, anything else that we study other than division, usually happens during this time. The next step in interphase is the S phase, DNA replication. New DNA is synthesized from existing chromatin. And so you have the chromatin that already exists. You got a strand of it. And so that strand splits and then copies get filled in for the parts that were taken away. And so the result is that you essentially end up, well, you had one, and now you fill in the gaps. Now you have 2 strands of DNA. So copied from existing chromatin, resulting in sister chromatids. And so that's the DNA replica. And it gives each cell double the amount of genetic information that it had previous to this step in interphase. That's important because when you divide, if you don't double the amount of information, when your cell divides, you end up losing genetic information, and that would leave you not human. G2 phase is the final prep for cell division. And so at this point, you have grown and done your normal process. You've replicated the DNA so that when you do divide, you can end up with the right amount of information. And then G2, the final prep for cell division, many of the organelles and molecules that are required for division are produced. We're going to look at the centrioles, we've talked about them a little bit, and the spindle fibers they'll produce. All that gets worked out during this stage. But that is interphase. It's the longest. We'll probably didn't take that long to cover going through it. I mean, most of the things that we study when we look at the cell are going to happen during this G1 phase. And then obviously S phase, DNA replication, that's the main point. And G2 is that final prep. Now there are, we'll cover this when I get back, there are checkpoints that happen in between these phases that prevent a cell from moving on to the next step in the event of an error. And we'll talk about the result of what happens if a cell finds an error, because that would be bad for you and for your cell. But we're going to keep moving through this. So once you've moved through the G1 phase, the S phase, and the G2 phase, the next step is the mitotic phase, or the M phase. And yes, that is for mitosis. So during this M phase, you have mitosis and cytokinesis. So I wrote mitosis down in green, cytokinesis in orange here. But for mitosis, remember PMAP. I'm sure you've heard it before. It helps you remember the order of the stages for mitosis. You got prophase, metaphase, anaphase, and telophase. So here's interphase. This represents everything that we just looked at. And so the cell grows, it replicates its DNA in the centrioles and gets ready for division. So you've got your nucleus intact, your chromatin is inside, and your centrioles are offset, still close together near the nucleus. Obviously for this, I didn't draw any other organelles, but you've got the cell membrane, the nuclear membrane, the nucleolus, chromatin DNA there, and the centrioles. And watch what happens to the nucleus and the information as we move through the steps. That's the whole point of mitosis. And so after interphase, the first step of mitosis is prophase. So the chromatin will condense into chromosomes. So you see the little squigglies, single squigglies. And so here we have kind of folded over into that standard X shape that you typically see with chromosomes. And so the chromatin condenses into chromosomes. The nuclear envelope breaks down. And so we once had an intact nuclear membrane. It now has gaps inside of it. And the centrioles are moving to the opposite side. So they were sitting here. Now they have moved from this position to, they're starting to move to opposite sides. And the spindle, which is like a web-like structure, it's microfilaments that are coming out that they're going to attach to the chromosomes to pull them apart. But those are starting to come out from the centrioles. And so one of the easy ways the amoeba sisters say it best, is the way you can remember this, is pro means to come before. So pro is the first step of mitosis. For metaphase, once this spindle reaches out, so once the nuclear membrane fully disintegrates, the spindle reaches out and attaches to the chromosomes. And you might see it on one of the worksheets we do, it might talk about prometaphase. Prometaphase is just a step between these two that some books will refer to, where it talks about that actually where the spindles reach out and they attach to this central point called the centromere. But I have it attached here in metaphase. So the chromosomes line up in the middle of the cell. The spindle attaches to the centromere on the chromosomes. You can think of M for middle. So you got metaphase, it's lined up in the middle. And then that leads us to our next step. Remember, if you need to pause, do so, because I know that you have to write pretty much all this for the notes, if I remember right, but you just have to write the name for that. I tried to be nice there. After metaphase is anaphase. The sister chromatids, which is the half portion there, are separated. So remember, we doubled up. So even though each side's only getting half of the chromosome here, because we doubled up during interphase and made those sister chromatids, each side will get the same amount of correct information. So anaphase, sister chromatids separate and are pulled toward each side. Easy way to remember this one is A for away. Anaphase, away. The last stage here is telophase. And so the chromatids will become chromatin and the nuclear envelope reforms. And so you have the chromatids, they unwind, become back in their chromatin state. See, same as they were in the beginning. And so once the chromatids become chromatin, the nuclear envelope begins to reform around the material. Your nucleolus reforms. The spindle dissolves, and so our centrioles are on the opposite sides. There's no spindle coming out of them. And the cell will begin to pinch in for animal cells or build a plate wall in plants. And so it happens differently between plant and animal cells. In animal cells, you get an actin filament ring that's actually going to contract, and it'll essentially squeeze in and pinch through the cell, separating it into two new cells. In plant cells, because they have a thick cell wall, it can't squeeze through it. What's going to happen is in that space, that rectangular space, they'll just actually build a new wall and make new cells. So that's one difference. One of the differences we'll talk about between plant and animal cells is the animal cells will actually, during telophase, they will have that cleavage furrow, is what that's called, and pinch in with those actin filaments. When I'm looking at this, because you will have to know what these look like on a test, I always think when this is pinched in, it favors an old telephone where you got the head, the receiving end, and then the speaking end on the other, and you got the two round pieces with this, it gets slimmer in the middle. You may not see it, but it helps me, so maybe it'll help you too. After we have done mitosis, PMAT, the next step is cytokinesis. And if you're like, wait a second, I thought this was the cell cycle and division. PMAT only deals with the splitting and dividing of the nucleus. Everything else gets divided in cytokinesis. So the word here means cytocell kinesis, movement. And so there's literally like a moving splitting of the cell here. And so cytoplasm and other organelles divide with the cell. The results in two new identical daughter cells. That's important because you don't want genetic variation here. If your stomach cells are making new stomach cells, you want the stomach cells that you just made to be like the ones that you previously had. You want to make identical materials to replace the old worn out ones that are leaving eventually, or ones that'll get worn down. So that's important to make two new identical daughter cells. Whenever there's a mess up or things don't happen and the DNA gets messed up, that's called a mutation. Unlike fiction and in comic books and movies, mutations are not good things. So you're not going to get superpowers. You're probably going to get really sick and end up in the hospital or with some kind of disease. So not a good thing. You want your cells to prevent any kind of mess ups. And so as I said before, there are these checkpoints I'll use this diagram. So there are different checkpoints that as it goes through, it actually will check to make sure that everything's lined up right. And if not, then the cell has a couple of different ways it can handle that. One of the phases is called G0. And this is like a temporary suspended phase. It's not in the cycle. It's like the cell kind of gets pulled out of the cycle, and it gets your cell tries to repair itself. And if it can't repair itself, then the next step is something called apoptosis, and we'll get to that in a second. But those checkpoints help make sure that there's no error in copy, because there's a lot of DNA to copy. And the fact that you do this all the time, it's easy to make a mistake. So your body has systems to fight against that. Make sure you get cytokinesis here. If you missed prophase, you can get that too. Make sure that you're studying these pictures. I mean, you're going to see it when you do the work packet, whatever day you get to it. But you're going to have to know diagrams on the test. Part of this, like the hard part of this, which isn't really hard. If you're good at memorizing, it's not hard. Is you just have to be able to look at this, look at the actual phase and go, oh, that's prophase. Or if I show you a picture where there's one cell with chromatin, everything's intact, though, that's an interphase. You see them lined up in the middle, that's metaphase. And so that's part of what this is. And then I'll ask you, okay, well, if this is prophase, tell me what's happening here. Explain to me what I'm looking at. And that's when you'll have to come over here and be able to explain this to me. Because this walks you through what happened from, how did we get from here to this cell? So that's mitosis and PMAP. This gives you an overview and shows you how interphase and M phase all fit together. An important note is that animal cells, this is another difference. So not only do you have the difference here, animal cells pinch in, whereas plants build a plate wall. Another note is that animal cells have centrioles, but plant cells do not. Plant cells will instead use microtubule organizing centers, or MTOCs for short. So that's a difference. You'll see that on the test. Just a good test question. So make sure you highlight, circle, do something there. I put a star beside it, but half of you won't even read it. So the next thing is cell cycle control. So you're probably thinking, how does a cell know when to do this? What causes it to move from cytokinesis to G1? What causes it to start, or how does it know to move to S phase? How does it know to move to G2 phase? How does it know to start mitosis? Biologists have discovered a family of proteins that regulate the cell cycle called cyclins. And so this is very surface level for your sake, especially since I'm not here. But these cyclins, or these proteins, they enter in through the cell membrane, and then they move in and they release hormones, so to say. They release chemicals to the nucleus that then signal a change that says, hey, it's time to start division. So these cyclins then communicate as they move through the cell to the nuclear membrane, let it know it's time to start. But these proteins are very important for regulating this cycle. If they're out of control or they're not working, we'll talk about that in a minute, can lead to problems. So one important group is the external regulating growth factors, which stimulate growth and division. It's very important during embryonic development. The embryo is the child in the womb that's growing. So there's a lot of rapid cell growth during this time period, because you have to go from a single cell to a baby. And so this process is very much at work. And so it's important for embryonic development, but also for wound healing. If you cut yourself and you need to get, in order for that cut to get better, you've got to make new cells, because when you rip through the ones that were previously there, so now there's a gap where there used to be cells. And so your cells actually will grow back most of the time. Nervous system and muscular, still up for debate. Most people will say no, they don't grow back. So the whole thing about losing brain cells is kind of true in that sense. So take care of your nervous system and your muscle cells, because once they're really damaged, they can repair themselves, but you're never going to make new ones, so, like you would skin cells or others. Uncontrolled cell cycle regulation can lead to many problems and disorders, including cancers. That's what cancer is, uncontrolled cell growth. So cancer cells do not respond to cell cycle signals or cyclins. And so pretty much when they get turned on, they do not turn back off. And so when these cells start to regulate and grow and divide, they miss the point where they're supposed to stop. So cancer cells often end up forming a mass of cells called a tumor. Tumors can be cancerous, but not all tumors are cancerous. That's important to note as well. What causes this? A defect in the genes that regulate the cycle. And how do your cells respond? Well, one, you try to go fix that G0 phase. But if you can't fix the cell in that point, the next best thing is apoptosis, which is programmed cell death. Basically, the cell gets put to death to prevent it from growing and dividing more. And that keeps that cell from spreading misinformation to other new cells. And so it's just a way that your body helps to protect itself and maintain homeostasis. And so that is the notes. Hopefully you got everything. We're going to be rolling through it more when I get back, but we'll review this and then we'll move to meiosis. I think you've already taken your test by this point, hopefully, for Quizlet for mitosis. Your next test will be the next Thursday. Yes, next Thursday for meiosis. Make sure you check out Quizlet. It's still going to happen. Make sure you're reading chapter 5 in the book. Understanding evolution, we're having a quiz on that. Look at the board. It's there. Don't fall behind because I'm not there. Make sure you stay on top of your work. I'm not sure you're going to finish the worksheet packet that I'm giving you for this, and that'll be okay. Like, don't panic. That's not an excuse to not do it, but you should still be working. But if you're working and you don't finish, I understand. We'll finish that on Tuesday when I get back. And so if you don't finish, hold on to it. If you do finish, you can go ahead and turn it in. Any other work that you finish, make sure that you've turned in this week. Make sure it gets put in that folder. If it's not in that folder, then you didn't do it to me. So make sure it gets in there. Make sure you're handling your business. And I think that's all I've got for you. Hope that you guys have a great rest of the week, and I'll see you when I get back. So see ya.